Dear students, after studying this module, you shall be able to know what is erythrocyte or RBC, what is erythrocyte isoenzyme, what is protein polymorphism, what is isoelectric point, and what is the significance of protein polymorphism from forensic point of view. We will start with the introduction of erythrocytes. Erythrocytes, also called as red blood cells or RBCs, are the furthermost common kind of blood cells and the animal's chief means of transport for oxygen to the body tissues by the blood flow through the circulatory system. They take up the oxygen in the lungs or gills and discharge it in tissues by squeezing through the blood's capillaries. Every second, 2.5 million RBCs are destroyed. Though that is only 0.0001% of the entire 25 trillion cells, the RBCs must be substituted if homeostasis is to be sustained. The first individual to define erythrocytes was the young Dutch biologist Jan Swammerdam, who, to study the blood components of a frog, took help of an early microscope in 1658. Uninformed of this work, Anton van Leeuwenhoek delivered another microscopic description in 1674, this time giving a more exact explanation of red blood cells. Malpighi in 1666 also published interpretations of human RBCs some 8 years earlier, and Leeuwenhoek's 1674 periodical on explanations of round globules in his personal blood. Karl Landsteiner in the year 1901 issued his findings of the 3 core blood groups A, B and C, which he later gave another name as O. Proteins are the major constituents of erythrocytes. These are the big biomolecules, or also known as macromolecules, comprising of one or more long chains of amino acids. They carry out an immense range of functions in living organisms, acknowledged as a different class of biological molecules in the 18th century. The isoelectric point, at times also shortened as IEP, is defined as the pH at which a specific molecule or surface transports no net electrical charge and consequently will not travel in an electric field. They precipitate most readily at their IEP, and the characteristic that can be exploited to separate combinations of proteins or amino acids. Diverse allelic forms of genes code proteins that can vary somewhat in their amino acid sequence. This process is known as protein polymorphism. Certain proteins circulating in serum show noticeable polymorphisms with alleles that have adequate frequency variances to be of significance in blood typing. Haptoglobin was the most extensively used of the polymorphic serum proteins in forensic biology. Carbonic anhydrase or Ca was the first discovered in vertebrate erythrocytes. Now, moving on to erythrocytes or RBCs. Red blood cells contain most of hemoglobin, a complex metalloprotein comprising heme groups whose iron atoms provisionally bind to oxygen molecules in the lungs or gills and discharge them all over the body. O2 can diffuse easily through the erythrocyte cell membrane. Hemoglobin in the RBCs also transport some of the waste products such as CO2 back from the tissues. Nevertheless, it is also transported back to the pulmonary capillaries of the lungs as bicarbonate ions dissolved in the plasma component of the blood. Myoglobin, a complex associated with hemoglobin, actions to store oxygen molecules in muscle cells. A characteristic human RBC has a disc-shaped diameter of roughly 6.2 to 8.2 micrometers and a wideness at the thickest point of 2 to 2.5 micrometers and a least thickness in the center of 0.8 to 1 micrometer, being much lesser than most other human cells. These cells have a characteristic volume of approximately 90 femtoliters with a surface area of roughly 136 micrometers square and can swell in a spherical shape covering 150 femtoliters minus the cell membrane expansion. The grown-ups have in their blood approximately 2 to 3 into 10 to the power 13 or 20 to 30 trillion RBCs at any given time, encompassing around 1/4 of the entire cell number present in the human body. Females comprise of about 4 to 5 million erythrocytes per microliter of blood and males about 5 to 6 million erythrocytes per microliter of blood. Individuals living at higher altitudes with low oxygen tension will have more erythrocytes. These cells are therefore much more mutual than the other blood components. There are around 4,000 to 11,000 WBCs and approximately 1,50,000 to 4 lakh platelets in each microliter of the human blood. To complete a single circulation of cycle, the human RBCs need an average of 20 seconds. The red color of the blood is because of the presence of spectral properties of the heme ions present in hemoglobin. Human RBCs consist of nearly 270 million hemoglobin molecules, each comprising of 4 heme groups. The hemoglobin contains around 1/3 of the entire volume of the cell. This protein is in charge for the transportation of over 98% of the oxygen. The erythrocytes of a normal human adult male stock en masse about 2.5 grams of iron, representing approximately 65% of the total iron present in the human body. Next, we move on to the life cycle of RBCs. In human beings, RBCs are formed by a process known as erythropoiesis. They are formed from the stem cells which matures the red blood cells in about 1 week. Once they are on the verge of their lifespan, they are removed from circulation. In several prolonged diseases, the life cycle of the red blood cells is evidently reduced. Example, in patients who requires hemodialysis. Now, the aging of RBCs. A characteristic mature RBC exists for around 120 days in circulation. When matured, the cells will exist for about 100 to 120 days in case of a healthy individual and for 80 to 90 days in case of a full-term infant. As the RBCs lack nucleus in them, they are not able to divide themselves or manufacture new cell components, and because of which these cells disintegrate or degenerate owing to aging and damage. Next, moving on to breakdown of RBCs. Old and damaged erythrocytes are excluded from circulation by the subsequent ways. Of these, 90% are removed from the circulation by the phagocytic actions of macrophages in liver, spleen and lymph nodes. 10% of the old cells hemolyze during circulation. Macrophages play a significant role by consuming these fragments of cells, and the lysosomal enzymes break down these chemical constituents in vacuoles. The aging RBCs go through modifications in the plasma membrane, making it vulnerable to careful recognition by the macrophages and succeeding phagocytosis in the mononuclear phagocyte system. For example, spleen, lymph nodes and liver, thereby eradicating old and defecting cells and constantly purging the blood, also known as erythrocytes. This condition is increased in a wide range of ailments comprising sepsis, hemolytic uremic syndrome, malaria, sickle cell anemia, beta thalassemia, glucose 6-phosphate dehydrogenase deficiency, phosphate depletion, iron depletion or deficiency, and Wilson's disease. The excessive erythrocytosis is witnessed in red blood cells or RBCs deficient of the cGMP dependent protein kinase type 1 or the AMP activated protein kinase AMPK. Inhibitors of the erythrocytosis comprise erythropoietin, nitric oxide, catecholamines and high concentrations of urea. Now we will study about the membrane proteins. The membrane proteins are accountable for the deformability, flexibility and durability of the RBCs, allowing it to squeeze through the capillaries of about less than half the diameter of the red blood cells, that is 7 to 8 micrometers, and recuperating the discoid shape once the cells stop getting compressive forces, very similar to a rubber object. Many disorders are caused by irregularities of the membrane proteins. Now we will study about the blood proteins. First comes the polymorphic soluble protein markers. Here, as shown in the table, there is variant found in RBCs such as the acetylcholine esterase, the acid phosphatase, adenylate kinase, carbonic anhydrase, peptidase ACD, phosphoglucamutase, glutamate dehydrogenase, pyridoxine kinase, and we have shown the variants found in WBCs and the blood plasma proteins. Proteins are not entirely rigid molecules so as to perform their functions. These proteins shall in between their various related structures. In the context of these functional rearrangements, the tertiary and the quaternary structures are generally mentioned too as the conformations, and the conversions among them are termed as the conformational changes. These conformational changes are a lot made by the binding of the substrate molecule to the active site of the enzyme, or else the physical area of the protein that take part in the chemical catalysis. The proteins also undergo changes in conformations due to the thermal vibrations and when molecules collide with each other. The proteins can be easily distributed into 3 core classes, which relate with characteristic tertiary structures: the globular proteins, the fibrous proteins, and the membrane proteins. Nearly all globular proteins are soluble, out of which there are several enzymes. The second class of the proteins is often the structural, for instance, the collagen, the main constituent of the connective tissue, and keratin, the protein element found in nails and hair. The membrane proteins a lot help as receptors or offer channels for polar or charged molecules to pass by the membrane of the cell. Now we will study about the isoelectric point. The isoelectric point, at times shortened or abbreviated to IEP, is defined as a pH at which a specific molecule or surface has no net electrical charge and as a result will not drift in an electrical field. The proteins precipitate utmost readily at their IEP, a characteristic property that may be used to separate amino acids and mixtures of proteins. The amino acids give proteins their total net charge, and these proteins may be negative, positive, polar or neutral in nature. At the pH lower than that of their IEP, the proteins have a net positive charge. Above their isoelectric point, the proteins have a net negative charge. Hence, they can be separated accordingly to their IEPs or the overall charge on a polyacrylamide gel by the means or a method known as isoelectric focusing that uses the pH gradient parameters to isolate the proteins. The IEP is also the first stage in the two-dimensional polyacrylamide gel electrophoresis. The proteins precipitate best at their IEP and a property that may be exploited to separate out the mixtures of amino acids or proteins. Next, we will study about protein polymorphism. Many proteins circulating in serum show noticeable polymorphism with alleles that have adequate frequency alterations to be of utmost significance in blood typing. The haptoglobin or the HP was the most extensively used polymorphic serum protein in forensic biology. The first instances of genetically determined protein variation were detected in antigens found in the blood cells, the so-called blood group antigens. Defective proteins can cause different kinds of diseases such as the oxygen-carrying proteins, for example sickle cell anemia, connective tissue proteins such as Martian syndrome, and the osteogenesis imperfect, and blood clotting factors such as hemophilia A and hemophilia B. The diverse allelic forms of genes code for proteins which may perhaps vary to some extent in the sequence of their amino acids. This process is also known as protein polymorphism. If these differences affect the protein's net electric charge, the different allelic forms can be separated using protein electrophoresis. We can identify the genotypes of particular individuals for protein coding genes and measure the allelic frequencies in the population. Over the last 25 years, geneticists using this approach have discovered far more variations than was previously expected, despite the high levels of polymorphism discovered using the protein electrophoresis. These studies underestimate both protein polymorphism and the total genetic variation present in the population. For example, the protein polymorphism that does not involve charge differences is not detected. Furthermore, because the genetic code is degenerate, more than one codon for most of the amino acids, protein polymorphism does not reveal all of the genetic variations present in the protein coding genes. The genetic changes that do not alter protein structure may alter the patterns of the protein synthesis during development and can be very important to an organism. When all kinds of variations are considered, it is evident that most species have an enormous potential for further evolutionary change. Now we move on to the erythrocyte isoenzyme. Erythrocyte isoenzyme, more commonly known as multiple forms of enzymes, are enzymes which vary in sequences of the amino acids, however, catalyze the same chemical reactions. These enzymes generally show diverse kinetic factors, example different Km values, or various regulatory properties. The presence of isoenzymes permits the fine-tuning of metabolism to meet the specific needs of a given tissue or the developmental stage, for instance, lactate dehydrogenase or the LDH. Carbonic anhydrase Ca catalyzes the reversible hydration and dehydration reactions of CO2 and carbonic acid. Cas are widespread in nature, being found in animals, plants, and certain bacteria. 16 isoenzymes have been identified and characterized in mammals. Ca was first revealed in vertebrates' red blood cells. The RBCs of human beings were used in studies in the year of 1970s to isolate and acquire the first amino acid sequences of the Ca1. John Etzel did widespread studies on the kinetics of human erythrocyte Ca and also discovered the first competitive inhibitor imidazole. Present studies on carbonic anhydrase continue to examine the mechanism of the inhibitor binding and CA's role in preventing the ectopic cardiovascular calcification. The 16 CA isoenzymes have been defined so far in mammals. The erythrocyte CA, CA1, CA2 are well known. The CA1, CA2, CA3, CA7 and CA11 are systolic. CA4, CA9 and CA12 and CA15 are membrane bounds. CA6 is secreted in saliva. CA5A and CA5B are mitochondrial. There are also 3 acatalytic forms referred to as CA-related proteins, the CARPs. Next, we move on to forensic applications. Some of the proteins flowing in serum show evident polymorphism with alleles that have enough frequency variations to be of importance in blood typing. The transferrin or TA and the group-specific components or the GC were 2 serum proteins which offered significant possibilities and were becoming normally used just before the beginning of DNA typing. Nevertheless, haptoglobin was the most extensively used polymorphic serum protein in forensic biology. Haptoglobin is a hemoglobin binding protein found in the alpha globulin fraction of serum. The 2 alleles, namely HP1 and HP2, have various exceptional variants at each allele. The alleles are separated by electrophoresis on a gradient polyacrylamide gel by a technique wherein the concentration of gel varies from 5% at the top to 30% at the bottom, thereby giving an improved separation by molecular sieving. The haptoglobin is a reasonably good system for use in forensic serology. It is constant with stains and the assay is very sensitive, requiring hemoglobin screening process, for instance leukomalachite green, to visualize the bands by reacting them with the bound hemoglobin. The hemoglobin is, however, one more protein made from 2 pairs of the polypeptide chains. It has different variations, all of the same structure for one of the pairs of the polypeptide chains designated by alpha. The dominant form is the HbA, that is the hemoglobin A formed of 2 alpha and 2 beta chains and is found in human adult beings. About 2 to 3% of the human adult hemoglobin consists of a variant called HbA2, in which the beta chains are replaced by 2 delta chains. A more noteworthy variation in the HbF or the fetal hemoglobin, which makes of about to around 70% of the hemoglobin in the fetal blood. The fetal hemoglobin consists of a pair of gamma chains as a substitute of the beta chain. The HbF is rapidly replaced by HbA after birth, and only a trace remains by the age of 1 year. The detection of HbF in blood stain is an indicator of the fetal blood. The typical test is grouping process of the blood group. Now, let us summarize all that we have studied in this module. The membrane proteins are responsible for the deformability, flexibility and durability of the RBCs, allowing it to squeeze through the capillaries. Most proteins bend into distinctive 3D structures. At a pH lower than their IEP, the proteins carry a net positive charge, and above their IEP, they have a net negative charge. Proteins can hence be separated in accordance with their IEP. The existence of the isoenzymes allow fine-tuning of metabolism to meet the specific needs of a given tissue or the developmental stage. Some of the proteins circulating in serum show noticeable polymorphism with alleles that have adequate frequency variances to be of importance in the process of blood typing.